Hi, everyone. Just to remind you, I'm Miranda Melen. Today, I'm going to be talking to you about some research that I've been doing on Stinkwort response to disturbance and competition. And then I'm going to check in at the end on a little bit of an update for seed bank dynamics that we've been looking at. As all of us have been discussing, Stinkwort is associated with disturbed soils. We see it growing out of pavement, along roadsides, in gravel areas, and in some pretty rough, disturbed regions. But why is there this association? And then you might ask yourself, why do we care? So you can think of this if we compare two different models. Perhaps this association between stinkwort and roadsides is abiotic driven. Maybe it prefers soil conditions along roadsides. On the other hand, maybe it's more biotic based, and perhaps it can handle the abiotic conditions out in the land where there's vegetation growing in our plant communities, but it's a poor competitor. And so therefore it's found along those roadsides because it's trying to escape from competition. So there are management implications depending on which model we find. If we find that it's based on this abiotic, it prefers those soil conditions along roadsides, then we can focus on those disturbed parts of the habitat and control for stinkwort in those areas. If, on the other hand, we find that it is more focused on biotic components that they're trying to escape from competition, then we can focus on reducing disturbance in our vegetated areas and promoting competition. If we are thinking about competition, then we might start to ask ourselves, well, but what would be the mechanism of that competition? We'll come back to that. Of course, it could be neither of these models. And so we would have this null hypothesis that since we know that roadsides are spread vectors, maybe it's just time and it just hasn't spread everywhere yet. And if that's the case, then we just need to be prepared. So when thinking about this first question of maybe it prefers soil conditions along roadsides, we did two experiments, germination and a greenhouse experiment looking at stinkwort growth. We used two different types of soil, a soil collected from the field, and then an engineered fill, which is very similar to what you find along roadsides. This engineered fill is what's used when you're laying down a roadbed or in construction sites. Here's what we found. When looking at the germination, we found that stinkwort doesn't germinate better in engineered fill. And we also found that if you look at this graph here, that engineered fill, a stinkwort biomass is on the y-axis, and there's a very, very thin brown line down right by the 0 mark. Stinkwort does not grow better in engineered fill either. In fact, we found that the big difference here is that stinkwort really does well in field soils. So if you were hoping for that, maybe stinkwort just likes roadside soils, turns out that it does not prefer abiotic conditions along roadsides, and it does just great in conditions out in our vegetation. Darn. So let's start thinking about that second model of perhaps it can handle growing out in a grassland or a rangeland, but that there's something that might be deterring it from just moving through that area and is instead excluding it, and you're finding it along roadsides or more disturbed areas. So perhaps it's an escape from competition. So now we have to ask ourselves, how good of a competitor is stinkwort? Maybe I should be asking, compared to a common grass invader, how good is a competitor? Perhaps let's think about Bromus hortiaceus or Festuca perennis. What do you guys think? Good competitor? Bad competitor? Okay, so we're getting some thumbs down in the room. So we did a greenhouse experiment. And here's what we found. We grew stinkwort by itself. That's the alone plot. We grew stinkwort with Bromus, and we grew stinkwort with Festuca. And we found that stinkwort growth was strongly reduced when grown with either of the grass competitors. So it really prefers to be by itself. That tells us it doesn't do well with competitors. So then we said, okay, what about soils? We have that engineered fill and we have field soils. So same plot, but different experiment. Now we have, again, alone plus bromus plus festuca with stinkwort in each of those pots. And what we found was soil strongly affects competitive interactions. So everything does really poorly in the engineered fill. So the main driver here in where does stinkwort do well? It really likes good soils, just like everything else. And it doesn't do so great with competition. So then we wanted to know, well, that was a greenhouse experiment. What happens in the real world? So we took this to a field experiment, and we wanted to ask about the mechanisms of competition. We wanted to understand better, are we thinking about below ground competition, above ground competition? If it's below ground competition, are we talking about competition for water or competition for nutrients? Or if it's above ground competition, are we thinking about thatch from previous generation out in the grassland, or maybe competition for light from other species? So this is beautiful Blue Oak Ranch Reserve, where I did a field experiment, and we had different plots out there where we manipulated different types of competition. And here's what we found. On this, on the x-axis of this plot, we have grassland control. That's where those plots, we didn't touch anything. It was just the grassland that grew up. Then in the thatch removal, we removed the intergenerational thatch, the dead grass from the previous year. So that's changing the above ground competition. And then in the above ground removal, we clipped the vegetation really low around the stinkwort and kept that vegetation clipped low. And then in the above and below removal, we went out there and hoed the entire area before putting stinkwort in, and then maintained and removed all competition from those plots throughout the growing season. And lo and behold, what did we find? Well, we found that stinkwort really liked that above and below ground removal of competition. But another interesting thing about this plot is I've got boxed the above and below ground removal. And if we compare that to the one right next door where we're just removing the above ground competition, what we're really seeing is that the important competition must be happening below ground. Okay, so we had these two models. We determined that the preference for roadside conditions was not true, but instead we're looking at that indeed there's some sort of escape from competition that is needed for stinkwort to thrive in an environment. Now I'm going to check in with you on our seed bank and germination behavior. This is a 5-year ongoing experiment, and we wanted to understand what happens to stinkwort seeds in the seed bank. We did 3 different experiments, a seed germination experiment. We looked at seed germination depth in the lab. And then we also have a field experiment where we're looking at seed dormancy and viability. So first, when we talked about, or when we looked at the seed germination, we asked, what happens to stinkwort seed germination over time? Here on this plot, we have months on the x-axis, and then on the right-hand y-axis is rainfall, and rainfall is in that kind of purpley columns. I don't actually want you to focus on the columns so much as just know that there was rain and then there wasn't rain. But what you should focus on is the green lines and dots. That's the proportion, the cumulative proportion of germination of stinkwort over time. And in the first year, right after that first rain in October, we found that germination jumped up to 65%. And by the end of year 1, we had 85% germination from these pots. So how we had these set up was these are open pots out in UC Santa Cruz, and they were getting the same light and rainfall and temperatures as any other stinkwort seed that might be found in a non-controlled area. But we were controlling to make sure these seeds didn't fly around, but they were exposed to open air situations. Now here's year 2. In year 2, we didn't see a huge jump. We found that instead germination was lower. In fact, only an addition of 1.8% germination occurred. Now, we're currently collecting year 3 data. That's ongoing, and it's raining nicely for us, and we're still seeing some germination. So I will be checking in with this group in future years when this 5-year study comes to a close. This is a great way of visualizing what we just saw on that graph. This is called a Sankey diagram. And here we've got a big green arrow, and that's showing the 85% germination. And then we can do some math out of 100%, and we know that, okay, 15% of those seeds are unaccounted for. With other viability tests that we've done, we calculate that about 3% of those seeds are probably dead, and about 12% are probably viable and ready to do something in future years. And just to remind you, the seed bank are seeds from the soil, from ecosystem that have mixed into the soil and are often just sitting dormant, awaiting germination cues. So in year 1, we saw that, and then in year 2, there was another 2% at the soil surface for these seeds. But we know that seeds move around, and we've even talked about how seeds can get buried, and we don't know what's going on there. So we really wanted to better understand what happens when seeds, stinkwort seeds, are buried. So we did an experiment where we put stinkwort seeds at 6 different depths in moist sand, and we watched them germinate. In fact, Emma, sitting in this room, watched them germinate for us. And what we found was sitting on the surface, as expected, we got about 70% germination rate, but there was a dramatic drop-off with depth, so that by the time you have 1 to 2 centimeters of sand sitting on top of these seeds, you have only about 10% germination. So now you should be asking yourself, what's going on with those seeds that haven't germinated? What are those seeds doing that have been buried? That's what we're doing in our 5-year seeds dormancy and viability field test. This is a lengthy process where we put 43,620 seeds, counted those out into seed bags. We have 4 time replicates. And every year we go out and we remove one of the time replicates, and we take those seed bags back to the lab and we count out all of the seeds that are remaining. And then we take those seeds that are remaining and we put them in a Petri dish, and we add gibberellic acid to break dormancy to try and get them to germinate. And that tells us, these were just waiting, they were sleeping, they're ready to germinate. Now, some of those seeds don't germinate. And so then what we say is, okay, which ones are, maybe those ones are dead, and that's why they're not germinating, but we need to find out if they're alive or dead. So we use tetrazolium and we do some staining, and we look for them under the microscope. And this is a picture of dead seeds. So we're able to get a really good snapshot of what's going on. So you remember that Sankey diagram that I showed you? This was when we looked at seeds on the soil surface. Here's what happens when you bury those seeds. So we see 15% germination. We see that dramatic reduction. Of those, we saw 20% of the seeds were dead, but 65% of those seeds were still alive. And when we dosed them with gibberellic acid, they germinated. So that's what's sitting in the seed bank waiting to germinate. We did see another 8% germinate in year 2 when we pulled up year 2 data. Haven't pulled up year 3 data yet, but that's coming. The conclusions from this is that most germination it happens after the first big rain event, which we know from Rachel Brownsee's work, and was held true here, where year 1 germination was at 85%. And in year 2 germination, we had a much reduced, only 1.8% that occurred. We know now that even slight burial enforces dormancy, and of those buried seeds, there's a high survival rate. So we had 65% after 1 year. Again, 5-year study, we're only seeing years 1 and 2. We've already talked about how 3 is this kind of gray area where we don't really know what happens next. But I will have results in the next couple of years. With that, I'd like to thank everyone that has been helping with this research, as well as funders, and especially the USDA, which has been funding this research. Thank you so much, everyone. Oh, and you'll need to speak through the microphone for everything.
So
there are no questions from Zoom right now.
okay, great. Can we have a question from the room? People are digesting.
That was a lot.
Great.
Rachel.
Wait.
Oh,
So you noticed that it likes good soils. Do you think that that means that we can worry less in low nutrient soils? I know I haven't seen it much, say in Serpentine, but we have a great room of people working in a lot of places. And
I know it's in Serpentine. So I can't say that you can worry less, just we know that it's going to be in disturbed soils. We know it's going to be along those roadsides. So those soils, there's nothing that's going to particularly attract them to that. They would prefer to be in more nutrient-rich soils. But if they are excluded, you're going to find them in those poor soils.
Just a comment. I kind of would like to see a that figure you had with the roadside fill or the engineered fill versus the field soil, where we saw the comparison of the three with the competition without. I'd like to just see the just the engineered fill blown up so you could see the three different alone versus the other two, because it does look like to the point of why it does well. And it's, I guess, what we probably all know, but that alone in engineered fill, it still can do even though it's not preferred habitat, it still does quite a bit better.
Yes, that's true. If you were to zoom in here, there is a slight difference in those almost to the 0 bars, and the alone is ever so slightly higher, and the error bars do suggest that there is some interaction going on there. But the big takeaway was the field soil. Yeah.
I would also like to see if you did something similar in sort of like, instead of engineered fill, but more of a gravel or sandy medium.
Ooh, that would be really cool.
Hi, such a great presentation. My question is how long you're going to hold this experiment?
The 5-year one?
5 years, just 5 years.
Just 5 years. And I mentioned that we
just
have
is not just.
so we've pulled up year 1 and year 2 data. This year is year 3. We will then take a break because I only had 4 time replicates. And so year 5 will come up in 2026 and we'll see what happens. So there's kind of like a stretch there. We won't know what happens in year 4, but we'll know what happens in year 3 and what happens in year 5.
And since your data, this is my next question, show the direction that, I don't know, to me, it shows, do you consider testing also additional things that you have to apply on top of the seeds? Because it looks like the depth plays it all.
Are you talking about different types of material, like
Yes.
adding mulch?
Yeah.
So we only did it with sand. Yes. I don't have any plans to. Of course,
Because
if
at
anyone
the
wants
same
to
time,
plant
you
some.
have, it looks like it's not only light, but it
Yes.
also it's water. So you may want to play a little bit with what can you add or how much. And then of course, logistically, what this will mean for a land manager, right?
I agree, Adona. There are a lot of really interesting questions we can ask different types of material that we all as land managers find that we're either putting on top of those areas or that it's growing out of. So whether it's different types of gravel or I've been up into the Sierra Nevada to see where Stinkwort stops growing, and it seems really strong right along the pine needle barrier. So that's another, mulch that's on top, but if we're using wood chips, it would be a very interesting study to try different forms.